Hi, and welcome to the AP Biology Lab 4 Plant Pigments and Photosynthesis podcast. I'm Mr. Anderson, and we're dealing with plants in this lab. So these are some plants, picture I took when we were in the rainforest of Ecuador. But if you look over here, this is a summary of photosynthesis. And so there are two parts of photosynthesis. Remember, there's the photo part, where you actually take light from the sun, gather electrons from water, but make NADPH and ATP. We then have the Calvin cycle, which we used to call the dark reactions. And what happens in the Calvin cycle is we take in carbon dioxide and we fix the carbon. In other words, we make it a usable form. And so we make something like sugar out of it. And so there are the two parts of it. Now, in this lab on photosynthesis, we mostly deal just with the light reaction side. But before that, I want to talk about chromatography. And chromatography you maybe did when you were little, if you would ever draw with a marker and then water-soluble marker on a piece of paper, and then as water ran through it, it would break that marker up into the different pigments that are found within that marker. Well, we do essentially the same thing. So what you do is you take spinach, in this case, we take spinach leaf, and then we use a quarter to grind that spinach leaf into a little bit of chromatography paper. So we use a spinach to grind some of that down in the bottom of it. So it looks, I don't know if you can see that, kind of like that. We then use a little bit of chromatography solution, and then we let it sit for about 20 minutes. And if you let it sit for about 20 minutes, what eventually happens is that spinach pigment is going to be broken down into different pigments that are found within the leaf. In other words, we're going to have chlorophyll A, we're going to have chlorophyll B, xanthophyll, beta carotene, so those are going to separate. Now, why are they separating? Well, they're separating according to a couple of things. If they really like the solvent, then they're actually going to stick with the solvent farther up. And if they really like the paper, chemically I mean, then they're going to grab onto the paper sooner. And so we can separate those out. Now, as they do that, let's take one for example. So like in this diagram, this would be chlorophyll B right here. The distance that pigment traveled, looking on the side, it looks like is 11, it looks like maybe we'll say 11 millimeters in this case. And then if you look at how far the solvent moved, that the solvent moved all the way up to maybe 18 millimeters. And so what's our RF value? Our RF value is going to be 11 divided by 18, which is like 0.61. And so that's going to be the RF value of that chlorophyll B. And so that's the first part, the chromatography, knowing that we can separate all the pigments in a leaf according to chemical characteristics. Okay, in this lab, what do we do? Well, in our lab, we use our hole punch again, and we punch holes out of leaves. We then put those holes inside, or the chads, we call them, that are punched out of the leaf. We put them inside of a syringe with some water, and that water also has carbon dioxide in it. You then build up a vacuum inside there, and what that really does in this picture is it takes that air spaces out and forces the solution into the spaces in that spongy mesophyll inside the leaf. We then put them in water and all of the chads will sink to the bottom. We then shine a light on it, and what's going to happen are the light reactions. In other words, we're going to convert that water into oxygen bubbles, which will build up inside the leaf, and eventually it moves up. The faster it moves up, just like in the enzyme lab, the faster the rate of photosynthesis in this lab. And so when we're done, we can analyze our results. And so in this lab, we do a number of different things. So let's say, for example, we're doing the intensity of light. Well, the intensity of light we can then put on the x-axis, since that's going to be our independent variable. And then we can measure the rate on the side. And so in our lab, remember, we always measure the rate as floats per second. In other words, if it takes one leaf chad 10 seconds to float, that's one float per 10 seconds, or we have a rate of 0.1 floats per second. Let's say something raises faster. Let's say one float per 5 seconds. That's 1 divided by 5, or 0.2 floats per second. And so that's going to be a higher rate. And so what do we find? Like in this case, as we increase light intensity, eventually we're going to increase that rate of photosynthesis. And we can use the slope to figure out the change in light intensity related to rate. Now, what are some other things we could do? We could do color, for example. Like what's going to be the worst color for a plant, it's going to be green light. That's what's reflected, and so we could find that maybe green light, if we shine it on it, it's not going to have a very fast rate at all. Or maybe the distance from the light source, or the temperature. Remember, all of this is molecules and enzymes, and if we can increase any of that kinetic motion or molecular motion, we can increase it. The amount of carbon dioxide we found is important. The type of leaf, obviously, or where you gather it on the leaf, those are all important. But remember, the dependent variable is going to be the floats per second, and that's a measure of this rate. Now, some people across the country use a different experiment, and they use a chemical called DPIP. Now, it's not important that you know how to do the lab, but it's important that you know what it is. So if you're ever asked questions about this, you understand what it is. It's just simply a chemical, and that chemical takes the place of NADPH. And so when it changes color, when it's reduced, it actually goes from a blue color to a clear color. And so you can use a machine like this. It's called a spectrophotometer, and you can measure the change from blue to clear. And the more quickly it changes from blue to clear, the more quickly the light reaction is going, and more quickly that DPIP is being reduced, or it's gaining electrons. And so that's another way that you could measure it. One important thing when you're doing this lab, we've done it in the past with mixed results, is that you have to use a heat sink. In other words, you put water in front of it, and the function of that is to make sure that we're not only changing the light in this reaction, but we're also getting rid of that heat. The heat is actually absorbed then by the water in here. And what you find in this experiment, as it's pictured here, is that if you boil a chloroplast, then it's not going to work. Or if you get rid of light, it's not going to work. But if we have unboiled chloroplasts, they're just going to start accepting those electrons, and we're going to see a change to the clear. So that's another way to do the lab. But regardless, you should understand what photosynthesis is and how we can measure the rate of it.